Okay, so in this presentation, what I want to do is to quickly go through some of the steps we've been taking in trying to take the PBPK, the physiologically based pharmacokinetic model that we've been developing, and move that towards a place where we can start to look at regulatory qualification of the model. So just as an outline, what I'll do is I'll talk about the regulatory use of PBPK models in other contexts, and then just to remind you, information we presented previously about the components of the PBPK model for TB drug development. I'll talk a little bit about the populations, the compound files, and the structure of the model. I'm not going to show a lot of the data today, but I will show one example with moxifloxacin, where we've been looking at making some predictions. And then lastly, to finish, just to talk about the strategy that we've been thinking about in terms of model qualification. So this slide's really looking at the regulatory acceptance of PBPK, and is going back to look at the submissions to the FDA with physiologically based pharmacokinetic models over the time period from 2008 to 2012. Now, just as a bit of historical context, the kind of idea behind physiologically based pharmacokinetic modeling was developed in the 30s, and it was used through the 1950s and 70s in toxicology field. It was really in sort of the mid-2000s that it started to be used regularly in the pharmaceutical industry. So what you can see on the left-hand panel is the applications to the FDA where they use physiologically based pharmacokinetic modeling in the IND and NDA submissions. As I said, this is 33 applications from 2008 to 2012. You can see that the main area of focus is the area that we were really working on at the time, in terms of predicting drug-drug interactions. But behind that, also looking at modeling the pharmacokinetics in pediatric population and a variety of other applications as well. The panel on the right-hand side is just showing that as well as taking models that were being submitted by sponsors, the FDA were also themselves looking at developing PBPK models. The other important thing over this time frame from 2008 to 2012 is actually we start to see the first examples where models that were developed in terms of physiologically based pharmacokinetic approaches were being used to inform product labeling. So there were first 3 examples where PBPK modeling actually appeared on the product label. And the example with ibrutinib is important because it was the first example where they'd used the PBPK model to do simulations and then use the simulations to waive actually doing some clinical studies. So that's the scenario from 2008 to 2012. There were 33 submissions. And really, this was a kind of a decade of work, really, to get drug-drug interaction predictions from really just a scientific question to really understanding more about model complexity, moving from static models to dynamic models, and then developing some of the concepts around verification and best practice, and really getting the models up to a stage where they could be used with used to answer regulatory questions. So there's quite a lot of work that was going on over that time period. What I'm showing now is actually the latest review of PBPK submissions to the FDA, and this covers the whole period from 2008 to 2015. What you'll notice is there's been 150 submissions just in the last 3 years. So it's really taken off in terms of the number of applications using PBPK modeling and in submissions to regulatory agencies. Still, if you look at the areas that are being addressed, it's predominantly around drug-drug interactions, modeling of pediatrics, and then other applications as well. But really, just to make the point that over that time period, there's been quite a big increase in the use of physiologically based pharmacokinetic modeling. This is now showing all of the drug labels where they've used physiologically based pharmacokinetic modeling on the label to make claims about the disposition and behavior of the drug. So now we're up to more than 20 examples where different drugs where this technique's been applied. And this includes 10 fast-track breakthrough priority approvals across a wide range of different therapeutic areas. And now, if you look through all of the data, there are more than 100 different claims that have been informed by physiologically based pharmacokinetic modeling. So in terms of regulatory acceptance, particularly on the drug-drug interaction side, then this is really moving forward quite quickly. As well as being used on the labels, both the FDA and the EMA have produced regulatory guidance in the last year. So the top's just showing the draft guidance from the EMA, which was issued in July last year and was followed up by a public meeting in November. And on the bottom, just the guidance on physiologically based pharmacokinetic modeling from the FDA, which was issued in December of last year. So at the moment, the EMA guidelines are being updated, and based on the meeting last week, it'll probably be towards the end of this year, or perhaps even the beginning of next year before we get to the final recommendations from the EMEA. So that's the kind of background to physiologically based pharmacokinetic modeling, its use in regulatory submissions. What we've been doing in partnership with Critical Path Institute was to develop PBPK models that we could use for TB drug development. So models that we've been developing on the SimCip simulator PBPK platform, and there'll be a demo hands-on exercise tomorrow if you're interested in seeing how that works. So what we're looking at are full-body physiologically based pharmacokinetic models, so you can simultaneously model a number of different compounds and metabolites, which obviously is important if you want to look then at drug combinations. And within those models, all of the moieties will mutually interact, so you can look at competitive mechanism-based and induction mechanisms, all of which will be important if you're looking at combinations. In terms of what we've been doing to bring together all of the information you need to model TB drugs, we've been looking at developing population files, so particularly around a virtual South African population, and I'll show some information on that on the next slide. We've developed compound files for the standard of care agents plus some of the newer agents, and also a multi-compartment lung model that I'll show you as we go through the presentation. So this is looking at the information we collected for the South African population, just shows how that we go around building up the populations that we use in the physiologically based pharmacokinetic modeling. So what we do is to kind of describe all of the covariates that we need to describe the behavior of the drug across the population. So here you can see the age distribution, and then once you've defined the age distribution, the age height, the height-weight distribution, and obviously once you know the height and weight, you can look at things like body surface area. The other kind of information we need to be able to parameterize the models, information around hematocrit, the protein binding, and obviously the different makeup of the individuals in terms of the genetics of the enzymes and transporters are important in metabolizing and transporting the drugs. So this is the kind of information that we collect together. We've also collected information that outlines some of the physiology changes that occur in TB infections so that we can account for that within the PBPK models as well. So we know about the weight changes and changes in BMI, but also some of the other things that will be important are increases in alpha-1 acid glycoprotein, which can affect the protein binding of the molecules and have influences in the drug disposition, and also the decrease in albumin concentrations that we know occur during TB infection. The hematocrit's also decreased. And obviously, if we're looking at disposition of the drugs into the lung, which is really one of the strengths of PBPK modeling, is really being able to get an idea or an estimation of the concentrations in the tissues, then things like how the lung pH is changing in different types of diseases will also be important. So that's the kind of information that we've been collecting together. This is just showing some of the simulations for the anti-TB drug, showing the simulated data as the lines with individual trials as the lighter lines, and the dots just represent the actual clinical data from those studies. We've also looked at the clearance across a population for a drug like isoniazid, where we know there are differences in the acetylator status that affects the pharmacokinetics. So again, the simulations that we have for that compound match pretty well what we know to be true from observed data. So I've talked about the population and the compound files we've developed. So this is a high-level physiologically based representation of a human. So what you've got is each of the compartments represents an organ in the body, and the organs are defined by physiologically realistic volumes and blood flows. Then if you look at administering the drug into the model, you can look at the drug distribution into each of the tissues and therefore understand how the drug's moving around the body and the concentration time profiles in the different compartments. What we've been particularly interested in doing as part of this project is to look at the distribution into the lung. So we've defined the lung by a permeability limited model where we've described each of the different lobes. So the different lobes are shown here. So each one of these represents a lobe. And you can see within the lobe, we're defining the alveoli, epithelial lining fluid, the tissue mass, and the capillaries. This is just showing predictions for one of the compounds. So this is just looking at the concentration of moxifloxacin in the lung after the compound's been given orally. And you can see that if you look in the left-hand side, the lung tissue to plasma ratio, you can see that using the model, actually the concentration in the lung tissue is reasonably well predicted. However, if you look on the right-hand side, you can see the concentration in the epithelial lining fluid for the compound, and you can see that there's quite marked under-prediction. And one of the reasons for that is in the model at this point, we were just considering movement of moxifloxacin to be passive. We know actually that it's transported by PGP, and the PGP is located on the apical surface of the lung, so we'll move drug from the lung tissue into the epithelial lining fluid. And therefore, if you account for the effect of PGP, then you should be able to get improved predictions of the epithelial lining fluid for the compound. And that's what we had to look at. So we looked at taking in vitro permeability data and extracting it and using it to calculate the transporter intrinsic clearance, which we scaled up to the whole lung. And you can see here, so this is the clinical data from 5 different studies. This was the data I showed previously when you don't account for the effect of PGP. When you account for the effect of P-glycoprotein, you can see the concentrations increase. The ELF plasma ratio is kind of in the area where the clinical data is, but you need a further correction factor to match the clinical data exactly. And that's one of the areas where we're still working in the physiologically based pharmacokinetic areas with the transporters. So what you see is when you look at the effect of transporters on drug kinetics and you look at doing in vitro in vivo extrapolation, at the moment there's a systematic under-prediction. So that's something in the field that's still being worked on. The last part of the TB model that we've been working on is a granuloma model to try and get closer to the concentration at the site of action. So what we've done is to develop a multi-compartment granuloma model that's shown on the left-hand side. So we're accounting for movement of drug from the rim blood into the interstitial fluid, into the macrophages, but also accounting for movement of drug into the casein compartments, which we've split in this case to casein 1 and casein 2. So the idea then is that you can start to model the drug concentration in the healthy lung tissue and in the epithelial lining fluid, but also within the granuloma. This is just showing some simulated and observed predictions from the model. So on the left-hand side, you've got rifampicin. So this is the predicted and the observed data in orange. For isoniazid, this is the prediction, and these are a range of clinical data. So these are 2 mean values, and then this is the range that's been reported in the literature. And then for pyrazinamide, again, the predictions and the range in reported values in the literature. So one of the problems or challenges with kind of the granuloma models having data to do performance verification against. But for the examples that we've seen so far, it's working reasonably well. And we've got some other data that should be coming through as well to allow us to do more performance verification. In terms of qualification of the model, what we've done is we've had initial a teleconference with the EMEA where we described the model and went through in more detail all of the different data that we'd collected. So we did that in September last year. And then we had a face-to-face meeting with the EMEA after their public workshop on PBPK in November. And so far, the feedback from the EMEA has been fairly positive, and we're just in the process of drafting some proposed context of use. So what we're thinking about for the proposed context of use for the PBPK models are 3 different scenarios at the moment. So looking at monotherapy, predictions of plasma, lung, and epithelial lining fluid prior to first in human, looking at doing similar kind of predictions in healthy volunteers and TB patients when you perhaps have some clinical data. And the third scenario would be around trying to do predictions across a wider set of compounds where we know that the compounds are affected by PGP, and therefore that will have an impact on the disposition of the compounds within the lung. In the discussions that we've had both as a team and with the EMEA, we did define some further context of use scenarios, but at the moment we've decided to not pursue those and just to keep them in the background. But it's something that we'll come back to later when we've got some more data. So just to summarize and to finish up, what I tried to show is that the use of physiologically based pharmacokinetic models in different regulatory scenarios has increased dramatically, particularly over the last 3 to 4 years. Based on the increased use of the PBPK models, and there have been some regulatory guidelines put forward from the EMEA and from the FDA, and the model that we've been working on over the past sort of 4 to 5 years, we're now in discussions with the EMEA and FDA about model qualification, so that we'll be able to use that to answer questions in TB drug development. We've defined different context of use scenarios, and as I said, we've narrowed those down to the most feasible, or what we think are the most feasible scenarios. So that was really the end of the presentation. Just like to acknowledge the people at Satara who've worked with me on this, and also obviously CPATH for their support and help as well. So if you've got any questions, I'd be glad to take them.